Guys, let's quickly do this. Please attempt these questions. The answer is behind. I'm just going to take this off. So just try to attempt the questions first before you see what I'm going to say. Eating food containing salmonella can cause illnesses. Two symptoms are vomiting, diarrhea. That's a revision in itself for you. In case you have forgotten the symptoms, and plus fever. Those are the major. Then, of course, plus abdominal cramp. Those are the examples. I mean, the symptoms, but that's not the question here. So, what causes these symptoms we've mentioned? One word. It could be, you could write one word. That's correct. Which is, you write toxins. If you just write toxins, you are correct sincerely. So, toxin released by the salmonella bacteria. But it's always good you write it this way because so there's a way GCSE likes to see the answer sincerely, so it's good you stick to that really. So moving on, give 2 ways the person with salmonella, now with mild salmonella infection, can help prevent the spread. So it's not as though he has it fully, but it is mild, so he needs to prevent this for that spread. So he can do that by what? 2 other ways: by isolating from others, all right, isolation from others, then practice hygiene to prevent, so by washing his or her hand with contact with things, and of course basically just to practice normal hygiene so that it's not going to spread for that to other people basically. All right, so the next question here says, in serious infection, now this is no more mild, a doctor can prescribe drugs to kill the bacteria. What type of drug should doctor prescribe? Don't forget it's bacterial infection, and that is what? Use of antibiotics. And this is correct for salmonella. This is correct for gonorrhea. Yes, because these are bacterial diseases. You can use antibiotics for either of this. Moving on. A person with AIDS may take longer than a healthy person to recover from salmonella infection. Explain why. 2 markers. Please make sure you attempt this question before you see my answer. So this is because the virus has destroyed the immune system. Anything you're going to say must reflect to the immune system. Anything you're going to say must reflect to the immune system. This is very, very important. All right. Then for this reason, the person no longer has enough antibodies, basically. Yeah, so don't forget what I just did here. So see the linkage. So no immune system, and what would the effect of that? Not enough antibodies to fight diseases. To fight diseases. All right. So moving on. Salmonella bacteria can be transmitted from chickens to humans. Chickens can be vaccinated to prevent the transmission of salmonella to the humans. So just one other ways farmers could prevent the transmission of salmonella from chickens. So what he's saying here is that, okay, vaccination should not be mentioned anymore because it's one of the ways. So what other ways can it be prevented? And that can be done by infected chickens should be isolated from other chickens or even killed. Yeah. Then don't sell infected eggs, so that let the whole thing stay there on the farm and it should not spread further. All right. Next question. A restaurant owner employed a scientist to test effectiveness of 2 kitchen cleaning liquid. The scientists look at the effect of took sample from the surfaces before the cleaning, that's here, then after the cleaning. So this was the result he obtained. So before the cleaning, both on sink and on table for liquid A. Before the cleaning, both on sink and on table for liquid B. Then after the cleaning for A and B. So we want to see the effect by looking at which one is more reduced after the cleaning. With what I just did now, you should know which one is more effective. Yeah, if you compare the after. So this is the answer here. From what we did, cleaning B is more effective. Like I told you, it's reduced more on the sink, reduced more on the table. So this is because it has greater reduction of bacteria after cleaning with B as compared with cleaning with liquid A on both locations. All right. Take note of the ways that those answers will be stated. The scientists investigated this effect of cleaning liquid A and B also on salmonella grown in the bacteria in the laboratory. So this is a salmonella that's being grown. All right. So what he's doing here is he has soaked filter paper into different things, into water, that's a control, into liquid A, liquid B. So after he finished, this is the result he got. So the circumference he saw, you can take time to look through these questions. I didn't want to because of time. So from here to here, for water, the filter paper that was placed in water, there was nothing that died. For liquid A, 11 centimeters square, then liquid B, 30 centimeters square. That means liquid B is more effective. That means, because what we're trying to do is that you look at where bacteria died around there, so you measure that circumference. All right. Now, to calculate, what you calculate is pi r square. But this that you have measured is diameter. All right. From your mathematics, you can tell this is diameter. So that means whatever it was here will be divided by 2 because this is r. R is half of radius. So if this was 4 millimeter, this whole place. So that means your radius will be what? 2 millimeter. Why? Because you have to divide it by 2. Hope that made sense. So it's going to be 3.14142 multiplied by 2 squared. Please don't forget that I only use 4 here just as an example. So the question says that, what measurement will the scientists need to calculate the area where no bacteria was growing? That's area, we call that area of inhibition, and that will be the area with greater size of inhibition. The scientists will calculate the radius of this area, basically. Yeah, that is the whole thing there. No, sorry, I'm not supposed to show that. Let's move on. So it says here that so give one other advantage, so one other change the investigation that will allow the scientists to check if the result is repeatable. All right, if it is something you can recommend, so or how effective it is. So what he needs to do is, if the experiment is repeated and it is consistent or the result is the same. So if it does the experiment again and still get the same value, then truly liquid B is truly effective. Next questions. It says here, now the scientists showed the results, okay, I already give it the answer to this one. Now metabolism is the sum of all chemical reactions. Wait, I feel like there's a question I missed here. Wait. There's a question I saw when I was calculating this. Okay, let's move on. Metabolism is the sum total of all chemical reactions in the body. So what metabolic reaction is the formation of lipids. Please, metabolism is all the chemical reactions happening in your body, both the ones where you are building things up like photosynthesis, or the ones you are breaking things down like respiration. So whatever you are breaking up or building up, that's metabolism in the body. So one of them is formation of lipid, which is you are building up. So what are the other examples you have? And that will be, okay, I knew it. There was a question I wasn't seeing there. Wait, why is it not here? There's a question that is hidden here. There's a question. Wow. The question is hidden somewhere here. Well, it's on the answer here. It says here, the scientists, please, let's quickly do this first, please. There's a question. I don't know why that is not in that other one. It's the same. Anyways, the scientists show the result to the restaurant owner. Both cleaning liquid cost the same, of course. So suggest one other factor the restaurant owner should consider when choosing this liquid, because the thing is this: in as much as B is more effective, he needs to think about, does it have any harm? I mean, it's going to harm the people. It doesn't have any other effects. If the side effect is more than the advantage, it's better it goes with liquid A, so that it's not going to claim people's life. So he needs to look at the side effect of this liquid B, or just of the liquid generally, both of them, and also see if it affects if it has effect on the cleaner's health. So if they inhale it, we hope it's not going to give them lung cancer or anything with time. So those are the things you need to look at. All right, so let's go to the next question, which we saw the other time. So it says that metabolism is the sum total. I don't know what really happened to this other file here, but I will find out later. So give one other chemical reaction in the body. Well, respiration, protein synthesis, which is formation of protein, glycogenolysis, which is breaking down of glycogen, glycogenesis, which is formation of glycogen, then photosynthesis. All of these are examples of metabolic reactions. Please don't forget that. Now here, table 2 shows I'm reading this question right now. So these are the ages of different people. Then these are the gender, male and female. Look at the mean metabolic rate for each of them. So what is your conclusion from this box? You have to look through this, pause the video and try that now. So the answer is, what I saw was that males have higher metabolic rate after the age of 5. So if you look at this, age of 5 for male and female was the same, but for female it was 45, 39, while that of female just dropped to 42, then 35, 35, 35, that way. Then secondly, I saw that the mean metabolic rate of females decreases faster, from 53 to 42 to 35. Then while that of male is gradually. All right. So those are the two conclusions I got from there. Next question. Calculate the percentage decrease in the mean metabolic rate between age 5 and 45 of so decrease in change. That means it's looking at these. It said between ages 5 and 45. So you are concerned with this number, these and these for the male, because that's what he said. So let's minimize that and see what is done here. So 53, if you look at the table up there, you take away 36 divided by who? 1 second. Divided by original metabolic rate, which is what? 53 still. So it's like original minus the final one. That way. So multiply by 100, that gives you this figure here. And if you round that off, it gives you this. Please, you can rewind the video if that was a bit fast. I'm trying to make sure the video is not too long. All right. Sorry about that. Then, regular exercise can increase metabolic rate. 2 people did 5 minutes of gentle exercise. Then, table 3 shows the effect. So these are the time in seconds, within 5 seconds, 3 seconds. Minute, please. 4 minutes, 5 minutes. These are the heart rate. See the heart rate. So describe 2 differences in the response of person R to person S. Looking at this here. So if you look at that, so you're supposed to study this. Pause the video here and attempt what you're seeing here. So what I saw, which is correct, is that the rate at which person R heart rate increase was slower. Please, was slower. If you look at this is 76, 85, like that. Then this one, it just kept increasing. All right. Then the second that I see here, the person heart rate of person R became more consistent. You can see from 85, 91, 99, still 99. That's quite consistent. But this one just kept going like that. All right. So the person S might have issues sincerely for that result, but that's not added here. Now, complete the line graph in figure 3 for person. You should add the scale to the x-axis and label the x-axis. So this is your y-axis, apparently this is your x-axis. So you're meant to label this axis. That's it. How many marker? 4 marker. Wow. So you label this axis, what should be here, then what are the values of seconds or minutes you put there. All right. So okay. So this is what I did. I also meant to plot the graph. So you're going to plot, where is that table? So for the which one was plotted? Let me see. Person R. So you're supposed to plot for S. So I'm going to put this aside so I can see. Okay, no, let me put this aside. Okay, I don't know if that is big enough. So let's see. This is my own graph here. Sorry, this time should be in minutes. Yeah, I think it's in minutes. Because what we have here, Abi, minutes. Yes, please, minutes. You have to be careful. Minutes. Then, so what do you need to do? Person O, sorry, the time at rest, the person's heart rate was, for person S, was 78. So what do you do? Let me shift that a bit more. Good. So from here to here is 20. So that means from here to here is 10. That means each box is 2 unit, as it were. All right. So that's why. So if you take this person at rest, it goes like that. 78 there. All right. That's what I just done there. I'll just do 2 of them. You see the remaining one. Then at 1 minute is heartbeat was what? Heart rate was 100. So if I take that up, walk with me. So from here, 100. Can you see what I did? That way. Then if at 2 minutes, his heart rate was 110. So look at this. I'm going that way. 110. So that's what you're supposed to do to plot this whole graph. And afterwards, you're meant to draw the line graph, which I just did here. So if you did all of these things, you would have your full mark, as it were. Questions like this, you have to be calm, all right? So that you won't miss out what the answer is. Moving on. So I think we've done that. It now says that we've done this. Calculate how much time it will take the heart rate of person S to return to its resting phase. So what do you need to do there? Now, you need to do what? Maximum heart rate minus, you take away minimum heart rate divided by heartbeat. The maximum beat per minute, which is this, you take away the minimum divided by the 12 beats, because that's what we were given here. This beat and heartbeat was given here, but these ones are beats per minute. Please, that's what I should have said at the beginning. Sorry, I should calm down. I think I'm rushing here. So the beat per minute is 132 beats. That's the maximum. Then at rest, the beat per minute was 78. All right. Then the heartbeat is 12 beats, so to say. So you input this value. So that's why I wrote it here. So this 78 from the table is up there, which I just showed you. Then this 132 is what is here. It's also on the table anyways. Then this 12 bits is from here. So if you do that nicely, this is what you get. Oh, if that was a bit confusing, just rewind the video quickly. A student made the following hypothesis about the heart rate of smokers and non-smokers. Now, this is what I was talking about. During the exercise, the the heart rate of smokers increases more than the non-smokers. So it's an hypothesis. That is his guess. So he just feel like people that are smokers, that their heart rate will increase more than non-smokers. So now design an investigation that would allow you to test the hypothesis to know whether it is true or not. So this is a 6-marker question. You are meant to, can you see the space? You are meant to write as much as you can. All right, so let's do this. So this is where I told you that you have to make sure you are writing your answers, to the maximum that you can. All right. For example, this question here, this kind of question, they always have level 1, level 2, level 3. Level 1 is always maybe just 2 marks. Level 2, maybe you have 4 marks here. Level 3, you get 6 marks or 5 to 6 marks, or sometimes you may even get up to 9 marks. Some books, some examination, some marking scheme will tell them to add more marks if the student has logical. I've seen that before. All right. So what you need to do is this. 2 experimental groups should be identified: smokers and non-smokers. Equal number from each. Maybe you say 10 smokers, 10 smokers and 10 non-smokers. Please, it's good to mention numbers here. Now, preferably, they should be the same age. They should be roughly the age should be the same across the groups. Heart rate of both groups should be recorded at the beginning. Yeah, so you should see what happens before the beginning. Then each group do the same exercise. So because you have to be, this is science. You have to give them the same task. Everything should be equal, because if it's not equal, that unequal thing can affect your results. So if you are using, for example, you use some groups are male and female, some groups are just males, then there's going to be bias, because that male factor and female factor will be something. And are they older or younger? Those are the things you should consider. So when you look at all of that, so you compare the result from each of the groups to show you the hypothesis is, to show if the hypothesis is what? Correct. So if it is not correct, you discard it. That means it is lie. It is not something to hold on to. But if you check it and it's actually more, then the hypothesis is good. All right. So take your time now to read through these answers. All right. So that's why I just zoom it that way. Next question. The circulatory system is composed of blood vessels, and heart. Urea is transported in the blood plasma. Name 2 other group substances that can be transported in blood plasma. Please, blood plasma is the non-cellular component of what? Of blood. In your blood, you have cellular component, which is what? Red blood cell, white blood cell, and platelets. This is one that carries oxygen and carbon dioxide. This one is what fights your infection, that defends your body, and this is what makes your blood to clot. But the water aspect of your blood is plasma. That's why you have all of those things. So the answer to this question will be that carbon dioxide, glucose, oxygen, water, amino acids, lactic acid or lactate, drug, hormones, carbon monoxide. I mentioned as much as possible. It was on purpose so that you can be able to remember. Now, do not write nutrients. Nutrient is vague, because when you say nutrient, you are not specific. So that doesn't, all of this I've mentioned, this is nutrient. This is nutrient. Yeah, so when you say nutrient, what are you saying? There are many more answers, many more. I can mention up to 50. So there are many more answers. All those ions, sodium, potassium, all of those are carried in the blood. So that's why you can see hormones. You know how many hormones we have? So those are the things. I wrote more answers here. So remember, these are the things present in the blood. Moving on. Now, some athletes train at high altitude. Training at high altitude increases number of red blood cells per centimeter cube of blood. Explain why having more red blood cells per centimeter cube of blood is an advantage to an athlete. 3 marker. All right. Okay. This is because the increase in number of red blood cells increases the amount of hemoglobin. What will hemoglobin do? More hemoglobin carries more oxygen in the transport system, which is what they need. So more oxygen allows for more aerobic respiration for the tissue and cells, thereby giving more energy to the athlete. Can you see the sequence? I told you, always don't just give one answer and just go. That sequence. So when you are studying your notes, make sure you understand it, all right? And be calm in your exam so that I can be able to arrange it, because if you are too fidgety, you might not be able to arrange your points in the right order, all right? So it says here, I want to stick to this. Oops, I already answered here. Okay, wait, I hope. Wait, okay, I think I was solving this with some questions, student in class. Which two blood vessels carry deoxygenated blood? That means blood that is poor in oxygen, and that should be pulmonary artery. So this is the only artery that carries deoxygenated blood. Other arteries carry oxygenated blood, and vena cava, please. So vena cava, of course, those are the big veins that come into the right atrium. All right. Moving on, I think I have a video on that on YouTube. You should check that up. Now, figure 4 shows 3 types of blood vessels. Which type of blood vessel carries blood to the right atrium? So the question I want to ask yourself is, what type of blood vessel goes to the right atrium? Is it vein? Is it artery? Is it capillary? And you should ask yourself which of them, what are these ones you have on the screen? So, of course, I took out time to label this out for you. So, this is an artery with a thick elastic wall or muscular wall with a narrow lumen, vein, thin wall or muscular wall. It's also elastic a little. It's a very wide lumen. And this is just one cell thick, and that is the capillary for gas exchange in and out of the thing. So which type of blood vessel will do that? That would be B. B is a vein. Now, if you look at your heart, what brings in blood to the right atrium are the what's that guy called again? The vena cava. I say vena cava because that's plural. Superior vena cava and inferior vena cava. Those are the 2 blood vessels that brings blood, and they are vein. That's why. So this question, if you don't even know what this is, you can't get the answer. So this is a vein, and specifically the name of that vein is the vena cava, which was what we mentioned here. All right, moving on. So you are asked to do what? Compare the structure of an artery with the structure of a vein. This is very important now. You can write as many as possible. So here, artery is made up of thicker wall. Vein is made up of thinner layer of muscular fiber. Please, muscle, make sure you compare them by that. You can see muscle, size of lumen, narrow here, wider here. So, this makes blood to move at a very high pressure. So it is called a resistance vessel for this reason. But because it's having large space at lower pressure, so it's called capacitance vessel. So it carries more blood. They do not possess valve. That is artery, while veins possess valve. Even though there's a bit of exception here. The pulmonary artery and aorta, they have valves inside the heart, just inside the heart, but it's not necessary. You're probably right. That's why I didn't add that here. Let's move on. Now, heart rates are controlled by groups of cells called pacemaker. I'm reading quite fast. So I already answered the question anyway. It says, draw X on where on figure 5 to show the position of pacemaker. So here is the pacemaker there. So I mean, no, here. Already, that's the answer to that question. Anyways, and I think I already did that here. Yeah, and the name of the pacemaker is what? Sinoatrial node. All right, moving on. Sorry, I use this to answer some questions to a particular group of students earlier today. A patient may be fitted with an artificial pacemaker. What condition will be treated using an artificial pacemaker? Please let me say this. I don't know if I have this video on YouTube yet or not. Now, this pacemaker is what makes the heart to contract, that bum, 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 bum that it does. Yeah, it's like it produces electrical impulses, which will make this place to contract first, then this will contract next. But usually when people need pacemaker is because when their heartbeat has become what? Irregular. So that's the answer to this question here. A case of irregular heartbeat. When the heartbeat is not regular, they need a pacemaker, which will help them to regularize that, basically. Now, a student, this is a lot of questions. Please, I want to pause the video right here and read through, because I'm trying to manage time. All right, just read through this, pause the video, read through this, and look at the answer. So if you've done that, this is what it is here. It says here, another student suggested that the result for this was anonymous. So why do you think it is true, if you think it is true? So well, this is true because the mass of gained by egg 4 is lesser than others. So others, look at this and this and this and this. But this one, this is not even up to 2. So that means it's not consistent. Look at this. This is about more than 2, or in fact more than 3, or thereabout. So this is anonymous because anomalous, please. Anomalous means that it is faulty. It's not really, there's something wrong. It's not consistent with others. All right, that's why that is the answer. Next question says, explain why the masses of the eggs increased. We're here now. So what's the answer to that question here? It says, this is because, I don't want to say the remaining part yet. This is because the egg has absorbed water by osmosis. This is possible as water moves from the beaker, which is having lower concentration, all right, or dilute solution, to the egg, which is at higher concentration, which is hypertonic. The water passes through the semi-permeable membrane or partially permeable membrane. This is testing your knowledge on on osmosis. Now explain how the student who could modify the investigation to determine the concentration of the egg, of the solution inside the egg. All right. So what you should do is that the student should use different concentration of salt or sugar in the beaker. So that means it's going to have maybe different. Maybe it's going to be having, it's going to be having 10%. 20%. I'm just giving an example, just different concentration. Then you should be able to plot a graph reflecting percentages. So if you have those different values I just wrote here, then there will be different percentages. You can be able to draw something out. Should determine the concentration where the line crosses from 0 to negative mark. If you see what we did in class, if those of you in my class, remember I showed you a graph like this, that the value that crosses and come down here, let me write that here. So it is a graph. So if it goes like that, everyone around here means they lost their mass reduced. Everyone around here, this positive side, means they gain mass. So something like that. All right, but you can only do that if it has different concentration, because you're going to be having a place with concentration, another one with what? With percentage change, as it were. All right. Next question. Chicken egg contain calcium. Calcium are absorbed from the shell into the cytoplasm of the egg. Table 5 shows how about the concentration. Please take note of this. Now, this is inside the egg shell, we have 0.6. Outside, you have what? 2.1. Okay, so it's like you're having something like this: 0.6 inside, 2.1 outside. Okay. Now it says calcium moved from this shell into, wow, so it's moving like this. What does that suggest to you? It is higher here and it's lesser here, and it's still moving like this. I hope somebody is thinking of the answer right now. That is what calcium moves from area of lower concentration to area of higher concentration, and this is done by what? Energy, and this is referred to what? Active transport. Can you see the way the answer is stated? You are supposed to establish the fact that you know that this is low and this is high, and yet it is moving this direction. So that's why it needs energy to do that. So what is being tested here is your understanding of what? Active transport. Please do not forget this. Moving on. Plants can be infected by viruses, fungi, and insects. Aphids are small insects that carry pathogens. Figure 6 shows an aphid feeding from a plant. Okay, all right. So an aphid feeds by inserting its sharp mouthpiece, which is called stylet, into the stem of a plant. Give the reason why the mouthpiece of an aphid contains high concentration of dissolved sugars after feeding. This is testing your knowledge on xylem and phloem. That is what is being tested here, basically. That's what's being tested here. So, what goes through xylem, what goes through phloem? This is because the aphid has just fed from the phloem of the plant, because phloem transport what? Sucrose and amino acid. Yeah. While xylem transport, of course, water and mineral salt or ions from the soil. This one is from the leaf around the body. If I've taught you plant tissue and all those things, you remember this. Okay. Plants infected with aphid may show symptoms of magnesium deficiency. Magnesium deficiency include yellow leaves, stunted growth. Explain how a deficiency of magnesium could lead to these symptoms. How would magnesium deficiency lead to this? This is a very serious thing, but it's not hard if you just can think. 5 marker, and you don't want to joke with this kind of question. So look at the answer I've written here sequentially. This is because magnesium is needed to make chlorophyll. So chloroplast is the organelle, but there's some pigment inside it called chlorophyll, which is the one that absorbs sunlight. All right. So for you to make that chlorophyll, you need magnesium. Now work with me. Let's say magnesium reduce the amount of chlorophyll. Let's say chlorophyll will reduce the light absorption. Reduce light absorption will reduce rate of photosynthesis. Can you see where I'm making it? Therefore, reducing sugar manufactured. So reduced sugar manufactured, or reduced sugar reduces the amount of excess glucose which would have been used to make protein, which is required for growth. So, because usually when you have enough sugar, some of them can be converted into protein and also even into fat as it were. So because there's no enough photosynthesis taking place, then there will be enough sugar. If there's not enough sugar, there won't be enough excess glycogen or glucose to be converted into all of those things as it were. Hope that made sense. That's the reason why it is going to be yellow leaves and stunted growth. Yellow because there's no chlorophyll. Stunted because there's not enough protein for it to grow. Can you see the way it links? You can rewrite that part if it's not clear enough. Okay. So it says here, a farmer thinks a potato crop is infected by a virus Y, potato virus Y, PVY, I'll just call it that way. So the farmer obtained a monoclonal antibody test kit for PVY. So to make the monoclonal antibody test, to make the monoclonal antibodies, a scientist first isolates the PVY protein from the virus. Describe how the scientists would use the protein to produce PVY monoclonal antibodies. This is just your knowledge about monoclonal antibody. All right, so let's see how it is done. Please check out. I don't have a video on this sincerely, but I don't know if I can do this within the short time before the exams. But let's see how it goes. So here it says that so what do you do? The protein from because what you want to do in the monoclonal antibody is that you want to use a substance to create antibodies for that substance. What do I mean? When you vaccinate people, what I'm saying is this: when you vaccinate people, you want to stimulate their immune system to produce antibody for that disease. All right. So that's what something we are doing here, but just that you are doing it artificially, and you are not using. It's not really for a disease, so to say, or it depends. You can use for anything, anyways. because monoclonal antibody is only for diseases, it could be for anything. Now, the protein from the infected PVY will be injected. That's will serve as what? Antigen. So usually what we introduce is the antigen from the pathogen. For example, when you receive, let's say, COVID vaccine, what was put into our body was attenuated, which means weakened or dead form of COVID virus, and that will stimulate your immune system to produce antibody against COVID virus. I mean, I'm just using that as an example, right? I don't know if it works really that effectively anyways. Now, but this same thing is happening here. So the protein from the PVY is injected into a mouse. Usually it is a mouse we use for this. So the lymphocyte of the mouse makes antibody for what? PVY. Can you see? It makes antibody. So the spleen producing lymphocyte, because the spleen produces lymphocyte, is combined with what? A cancer cell to form hybridoma. Don't forget this word. All right. So the reason why you're combining with cancer cell is that so I can reproduce faster. The spleen cell will not produce enough lymphocyte which will produce antibodies on time. So you just need to be able to make it faster, because what happens is this guy is what will produce antibody. So, and you want this guy to divide fast, but it can't be fast except you add it to a cancer cell. And that cancer cell is called myeloma cell. So that myeloma cell is combined with the spleen cell to form hybridoma. All right. So that hybridoma helps to make many lymphocytes, which produces a lot of antibodies within a short while period. All right. Pause the video here. Read what I've said clearly, or I mean slowly. Now, cystic fibrosis is a genetic disorder caused by a change in gene. What molecules are genes made up of? So what is in gene? Or what are the genes? What are genes made from, so to say? And that will be your what? Deoxyribonucleic acid, which is DNA. All right. Moving on. CF affect the cell membrane of cells of the lungs and digestive system. That's cystic fibrosis. What is the function of the cell membrane? That's more like testing your knowledge on cell. It controls the movement of substances into and out of the cell. It holds the cell together. One of these answers is already good, but preferably this will be the first answer you should mention. Very important. Now, in a person with CF, cell linings in the lungs and digestive system create so much mucus. The mucus can do what? Block the duct leading from the pancreas in small intestine. That's not nice. Block the tube leading from the alveoli in the lungs. Man, that's not nice. That's why it can be so deadly. Explain why children with cystic fibrosis grow slowly more than children without cystic fibrosis. 6 marker. You don't joke with such question. This is testing your knowledge about. It's not even about this. It's not about cystic fibrosis here. It's talking your knowledge about digestive system and respiratory system, basically. And that's what is happening here. That's why I said these things are applied. So these are the answers. The pancreas is essential for digestion as it secretes digestive enzymes. So when the mucus blocks the ducts of the pancreas, fewer enzymes are secreted into the small intestine, which is basically is the duodenum. I have a video on that. Now, the decrease in enzyme reduces what? Digestion in the small intestine. Decrease in digestion leads to what? Less absorption of glucose and amino acid. And when this is reduced, these amounts of amino acid leads to what? Lesser protein necessary for growth. Can you see that linkage? You want to pause the video right now to see this whole thing. Of course, then on the other hand, blocked alveoli will decrease oxygen intake because the oxygen that will be inhaled will be lesser. So decreased oxygen will decrease amount of oxygen available for what? Respiration. This will lead to decreased energy necessary for growth. This is if you write this, I'm very sure you're going to get the extra 3 marks, which is for logical. So this is 6 marker, but because this was written in a level 3 format, it's going to have 3 marks to it. That's going to be 9 marks for real. Please don't forget this. see the logical linkages. That's why it's good you understand each of those things. Pause the video here to see the answers.